So I think that the main challenge here, but also what drives me the most, is how can you make transfer learning happen between those two areas of expertise. So how do you manage to have ML engineers that understands enough the bio so they can make the right decisions when developing the architectures and developing the models? But at the same time, how can you empower the biologists so they can be autonomous enough to answer their own research question using the tools that are being developed?
Hi, Marie.
Hi, Rebecca.
Thank you so much for volunteering to speak to me today. I'm doing a series about women in AI, and now I'm back in Paris, speaking to one of my colleagues in the Paris office. So we already know each other, but I know nothing about your field, so I'm really looking forward to speaking to you today. So I know that you're an expert in genomics. You did your PhD in the subject, and now you're a team lead at InstaDeep, a team that's working on this topic. So yeah, maybe you could explain a bit about what excites you about genomics and where AI fits into that.
Sure, absolutely. It's true. I've done a PhD in population genomics in the past. And so this was involving a lot of statistical modeling on how genomes of humans behave across time and in evolutionary times. So at InstaDeep now, what we're doing with the team is to try to apply AI techniques to the genome, and specifically language models, which make actually a lot of sense for genomics, as there is a tight regulation that happens in our genomes in order to make our cell functions. And language models have this ability to understand the grammar and syntax of strings. And this is exactly what we do have in our genome. We have strings of A, T, C, and G nucleotides. And the goal is to see if we can decipher a bit what's within our genomes thanks to these new technologies.
Oh, cool. So it's great that we're able to take technologies that have come from maybe, translating language, and now we're applying it to something completely different, because there's this same idea of there being relationships between, so a string of words in the case of language, but here it's a string of different types of molecules put together. So I know that your team's worked very closely with the research team at InstaDeep, and that you've recently published a paper about the nucleotide transformer. So we've heard a lot about transformers recently within the field of AI, but this is a specific one for genomics. So maybe you could speak a little bit about that.
Yeah, absolutely. So we've worked collaboratively with our research team internally, researching in NLP. And we actually, in a partnership with NVIDIA, got access to a lot of compute. So we got access to Cambridge 1, which is the largest supercomputer in Europe. And this enabled us to feed the model with billions of nucleotide sequences. And we actually took an approach where we trained models for humans specifically, but we also trained models with sequences that are coming from 850 different species, from elephant to bacteria, basically. And what we learned from that is that through the evolutionary history of all living beings, there is some kind of experiment that has been conducted without us knowing about what we can keep in a sequence to make it work, and about what we cannot keep in a sequence because it's too deleterious. And through this, the observation of this diversity that exists for the different parts of the genomics feature, we can actually learn what are the patterns that make a gene, or a promoter, and we can extract some generalizable features and apply that for human genome discoveries and human genome prediction, basically.
Okay, so with all of this background data from all these different species, so I guess when you're speaking about bacteria, it could be species that have existed for a lot longer than mammals, for example. The model's somehow learning things from that it can then transfer onto being able to predict things about humans.
Mm-hmm. Yeah, absolutely. And this is what we have observed also in the results of the paper. So we have trained this foundational model that actually gives us a high-level vectorial representation for any DNA sequence. So what it means is that the model is able to translate and to represent any sequence of DNA in the form of numbers of 1024 number vector. And after that, we use this vector and we assign labels to that in order to perform prediction on different downstream tasks. And we have evaluated the model on 18 different downstream tasks to predict regulatory features, for example, in human genome or in the genome of other species. And what we see is that we can actually outperform many of the models that were specifically trying to do a single thing. We can outperform most of those on the 18 tasks that we have evaluated the model on. So what it means is that the model is able to give meaningful representations that we can then use and apply in a very versatile way to different downstream tasks.
Okay, so just so that I check that I've understood, so you've got these 18 downstream tasks. Did you use the foundational model directly? Or was there any fine-tuning that you did on that foundational model for those specific downstream tasks?
Mm-hmm. So actually, in order to evaluate the model in the best way possible, we have carried on both experiments. So first, we have tried to see how the embeddings, so the vectorial representation of DNA sequences with the labels and just the logistic regression, were able already to give good results on classification tasks. And after that, we have also applied an efficient fine-tuning. So efficient in the sense that we are only updating 0.1% of all the parameters. And applying this fine-tuning, we're able to increase even more the performances and to outperform most of those models. And so even though fine-tuning is giving better results in general, we see that we can already reach great performances only with embeddings, which require basically zero compute or very little compute. So this is a tremendous help for all the different genomic prediction tasks that someone in the lab, for example, might want to carry on, because it's easily applicable and it can run on a laptop.
Yeah, that's really great. So it means that there's a lot of work and compute has gone into training the foundational model, and that doesn't need to be redone and using all of these resources multiple times. So you can be efficient in terms of energy usage and all these sort of things.
Yeah, absolutely. And it does not require a lot of machine learning training either. So it's readily applicable for the bio and genomics field.
Okay, so you can use this without being an expert in AI, which is nice.
Yeah. If you go to our Hugging Face repository, you'll see that in less than 10 lines of code, you can actually use the model and fine-tune it or train another model on top of that to perform the prediction that you're interested in. And it also requires a lower data setting. So thanks to this pre-training that has been done with a few thousand observations that you might have acquired through experiments in your lab, can definitely input that into the model together with the embeddings and already get a classifier that is specifically trained for your use case.
Oh, that's really great. And this is something that I really like about the AI research community, is that there's quite a lot of things that are available open source. So not only can people read this paper and learn about all of the work and training that you guys have done within the genomics team and also the research team at InstaDeep, but there's also the code there ready to go for people to make practical use of it, and the other teams can also further their own research with it.
Yeah, and I truly think that this is the way to go moving forward, to be able to have the whole community using tools that can be easily leveraged.
So you started off as a very technical person. So you've done your PhD and, you're very involved in the research, but you're also now managing a team that's grown quite quickly. How do you balance, the technical side and the managerial side, and what kind of challenges have come your way over the past year or so where you've been building this team?
Yes, so as you mentioned, this is a very technical area, so we need a great deal of expertise and we need many experts in both fields, right? In the biology field, genomics field, and even within this field, you have different areas of expertise. So you might be an expert on ecology or regulatory genomics or population genomics. And at the same time, you need people that are keeping up with the AI literature and being able to implement the latest research that is being generated at a very fast pace in the AI field. So I think that the main challenge here, but also what drives me the most, is how can you make transfer learning happen between those two areas of expertise? So how do you manage to have ML engineers that understands enough the bio so they can make the right decisions when developing architectures and developing the models? But at the same time, how can you empower the biologists so they can be autonomous enough to answer their own research question using the tools that are being developed. So I think that my main goal at managing the team is being able to have this happening inside the team, and to have this transfer happening, and to have both sides of the expertise that are collaborating in a way that we are sure that we're moving forward and we're bringing the best research we can to the table.
So how big is the genomics team now, and how fast did it grow?
It was a team of a single person a year and a half ago, and now we're 15 people working together in the genomics team. So yeah, it has grown pretty fast.
Yeah, and these are your team to be found all over InstaDeep's offices, or all in the same place?
I wouldn't say all of the offices because we have so many of them, but we have a majority of the team located in London. We have also team members in Paris, in Tunis, and in Cape Town at the moment. And we're also collaborating with the research team located in Boston.
Okay, so we're covering quite a lot of the globe there, which is nice.
Yeah. And inside the team, there is also like a very high diversity of backgrounds. So yeah, that's a very stimulating environment, and we managed to have a nice synergy happening.
And so you said that you went from size 1 to size 15 quite quickly. That must have meant that you were doing a lot of interviews.
Yeah. Yeah, that's for sure.
So what kind of things did you learn from, going from the technical and interviewing people and seeing whether they're a good fit for the team?
Mm-hmm. Yes, so there is definitely a learning curve here to know how to carry on the interviews and making sure that everything is going smoothly. I personally have made the choice to have a very technically oriented process for hiring. I think this is something that has been working quite nicely so far. So we have technical tests that are specific to different position, and the interview process always ends with the presentation of some project of the person has been working on to the rest of the team. So we can also all agree that we will be happy and thriving working with this person, and also for the person interviewed to be able to meet the entire team and be sure of the decision of joining.
Do you find that, so this podcast is about women in AI, and we speak a lot about, maybe there are unconscious biases in interview processes. Do you think that keeping it technical is one way of being able to get rid of some of those biases?
Yeah, definitely. You get rid of a lot of biases by focusing on the technical aspect. Of course, it's not the only aspect that has to be taken into account, as I said previously, but of course you can remove a lot of biases, and it's true for the gender bias, because the way that women and men present themselves or talk about themselves technically can differ.
Okay, so it's been super interesting to speak to you more about the research paper that you did, the work that you do day to day. Is there anything else you'd like to say before we go back to work?
I would say that it's been a great journey and a tremendous opportunity for me to be able to build a team and to hire people that I'm learning from every day, and to be moving in this space that is extremely exciting, because everything has to be done basically. So even though this is only the beginning, I'm very, very excited about what's coming next in this area. I'm sure this is exactly the time for AI to be looking more into what biologists have been making for the past 50 years.
Thanks a lot, Marie. And see you
Thanks
soon.
a lot, Rebecca.
Bye.